In this video, we will discuss about how the glycolysis regulate anchorage-independent changes in the PDAC-derived cells. In the previous video, we have discussed about the role of CD24 and CD44 in the anchorage-independent changes, that is, the growth rate and invasion properties from the PDAC-derived cells. In this video, we will understand the role of glycolysis in those characteristics. So the glycolysis or the glycolysis, how the authors come up to this glycolysis pathway is that during the transcriptome analysis of the derived cell lines, that is SP0926 and SP1030 and the other derived cell lines, and compare them with the mother pancreatic cancer cell lines, they find out that there is a lot of metabolic pathways that are upregulated. And so since the glycolysis also plays a role towards this metabolics pathways, they hypothesized that this glycolysis might be upregulated or dysregulated in those might be upregulated and dysregulated or plays a role towards these anchorage independent characteristics that are shown in the PDAC derived cells. So first what they did is they checked the expression level of this SLC16A1 expression levels in all the three samples, that is in the mother pancreatic cancer cells and the other SP0926 and SP103 derived pancreatic derived cell lines. So the role of this SLC16A1 is to help in this help in this glycolysis pathway. The SLC16A1 helps in the glycolysis pathway. So they needed to check whether this SLC16A1 is upregulated or not, and they find out that it is upregulated and upregulated compared to the pancreatic cancer cell lines. However, the difference between both these derived cell lines is not much, and these things they have validated using the genes that is responsible for this SLC16A1 formations, that is MCT genes. And in this case also they have find out that these MCT genes are upregulated. That is, these two genes are present in the case of this SP0926 and SP1030, and this is the main gene that is at 100 kilodalton. We can see that in the case of SP0926 and 1030 it is upregulated, and this is the actins act as a control. And thereafter they check the lactate concentration, which is the product for this glycolysis, in the cell culture medium and also in the cell lysate. And what they have find out that these lactate formations are high in SP0926 and SP1030, and also this lactate amount in the cell lysates are also high compared to the pancreatic cancer cell lines, are statistically significant in both the cases. Now they have understood that glycolysis pathways are upregulated. They wanted to check whether these upregulations of this lactate and this SLC16A1 is because of this glycolysis pathway or there is some other pathways. So in order to check that, they inhibited these genes. They inhibited these MCT genes and check its effect on this properties on this invasion and growth properties of the pancreatic derived cell lines. So they did it. The data are shown here for the 2 rounds, and they can see that in the case of SP0926 the tumorigenic properties are being reduced when they use AZD, this MCT1 inhibitor, that is AZD0365, and also this in the case of SP1030 in SP1030 the tumors formations are reduced, and in the case of SP0926 the invasion properties are reduced. So this is the first case, and this is the first round, and this is the second round. You can say that second replicates, where we can see that the invasionness of this SP0926 are reduced and the tumor cells are also reduced compared to the mock case. However, for the pancreatic mother cell lines, it does not have much effect on in both the rounds on tumor properties or on invasive properties, and the quantifications are shown on the right hand side, where we can see that the mock SP1030 has high tumorous pairs compared to the case of this AZD39036965 treated cells, and it is same for the second round also. And when they check the diameter of the tumorous pair, they also see the diameter of the tumorous pair also reduced in the SP1030 when they are treated with AZD09365 0965, that is the MCT inhibitors, and same results they have observed also in the second round. After that, what they did is they checked the effect of this AZD09365 in the pancreatic cancer pancreatic cancer cells and derived cell lines in the presence of DMSO, and compare it with this inhibitors, MCT inhibitors, and they can see that in the case of these MCT inhibitors, the reduction of this invasiveness is reduced, and also the at the same time this tumorigenic properties in the SP1030 cell lines are also reduced. The quantification of this penalty is on the right hand side, where we can see that in the case of SP0930 the invaded cells are less, and also for the case of SP1030 the tumorigenic cells are less, and but this difference is not statistically significant, and it's similar to the DMSO. So things that is significantly reduced is in the case of this invasion properties in SP0926. So this indicates that all these results indicates that the glycolysis pathways are been responsible for, or are dysregulated or upregulated, that is responsible for this anchorage independent changes in the pancreatic derived cell lines as one of the important component. And this DMSO results also shows that it is not the effect of some other pathways that get dysregulated. If you need to understand more about these experimental results and how they have conducted their experiments, what are the reagents they have used, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.